Genomic libraries in high capacity vectors. In addition to lambda vectors, we can also use other vectors that can clone even larger DNA fragments. From previous topics, you have the idea that different type of vectors are available that can accommodate comparatively larger DNA fragments. मिसाल के तौर पे हम यूज़ कर सकते हैं बैक्टीरियल आर्टिफिशियल क्रोमोसोम्स, देन P1 derived artificial chromosomes या यीस्ट आर्टिफिशियल क्रोमोसोम। तो इनका एडवांटेज ये है कि बैक्टीरियोफेज लैम्डा के कंपैरिजन में ये ज़्यादा जो बड़ा डीएनए फ्रैगमेंट है उसको ये क्लोन कर सकते हैं। तो व्हाट मे बी द एडवांटेज ऑफ क्लोनिंग इन vectors or high capacity vectors? The advantage is that the size of the genomic library that will be produced, it will contain comparatively low number of recombinant clones that is easy to screen. Again, I can repeat that what is genomic library? A genomic library, it represents the whole genomic DNA of a particular organism in the form of recombinant clones that also contain our gene of interest. अगर number of clones होंगे तो उसको स्क्रीन करने में ज़्यादा टाइम लगेगा, लेकिन अगर हम हाई कैपेसिटी वेक्टर्स को यूज़ करेंगे तो उसकी स्क्रीनिंग के लिए जो टाइम है वो कंपैरेटिवली कम यूज़ होगा. So this is one of the advantage of using high capacity vector. So another example of high capacity vector, it is the cosmate that can clone up to 45 kilobase of DNA fragment. Although it is very advantageous, here again the size of the genomic library that will be produced in comparison with lambda genome, it will be very small, because here the larger DNA fragment, it has been cloned around 45 kilobase. But it has certain limitations. Both vector, lambda vector and cosmate vector, they have certain limitations. Like in case of bacteriophage lambda, comparatively small fragment of DNA around 20 kilobase pair, they can be cloned, but in case of cosmate, comparatively larger DNA fragments it can accommodate. But the screening of colonies after plaque formation in case of cosmate vector, it is comparatively not, you can say that, so much suitable, because the hybridization background, it is less in case of cosmate vector. And hybridization we use to detect our gene of interest by using specific probe. Whereas in case of lambda genome or lambda vector, here the DNA hybridization, it is very clear. It gives clear results. So this is one of the advantage of bacteriophage lambda. Another advantage of bacteriophage lambda is that the shelf life of genomic library, it is comparatively longer as compared to cosmate libraries, because the shelf life of E. coli host, it may be you can say compromised as compared to bacteriophage lambda, because in case of bacterial host, the viability may be the issue. In addition to that, we can also use high capacity cloning vector. As I told you that bacterial artificial chromosome or yeast artificial chromosome, because they can clone comparatively larger DNA fragments. Like bacterial artificial chromosome, they can clone around a 200 kilobase DNA fragment, and in case of yeast artificial chromosome, तो जो DNA की length है, it may be more than 200 kilobase, like maybe up to 2000 kilobase. So larger DNA fragments they are produced. But during cloning procedure, there may be some difficulties because of the large DNA fragments. So it is not always preferred. These high capacity vectors, they are mainly preferred where the genome mapping is required, or the subsequent sequencing it is required for different type of source DNA.